The University of KwaZulu-Natal's College of Agriculture, Engineering and Science is committed to making a difference in our world, from the classroom and laboratory to the field. It provides one of the finest study and research environments on the African continent and is recognized internationally as a center of excellence. Agriculture, Engineering and Science is one of 4 colleges that make up the University of KwaZulu-Natal and is home to over 8,700 students and approximately 700 staff. 3 of the university's 5 campuses form the core of agriculture, engineering and science activities in the region. The college comprises 5 schools: agricultural, earth and environmental sciences, engineering, chemistry and physics, life sciences and mathematics, statistics and computer science. Engineering is offered on the Howard College campus, agriculture on the Pietermaritzburg, and all the other sciences on both the Pietermaritzburg and Westville campuses. Exceptions are agricultural engineering, which is offered in Pietermaritzburg, and geology, which is offered at Agriculture, Engineering and Science. We offer a wide array of interesting and innovative degree programs that open doors to a variety of different careers. Some degrees are unique to UKZN and are not offered at other tertiary institutions. For example, we are the only university in South Africa to offer a fully accredited agricultural engineering degree and one of only 2 universities in the country to offer a BSc in land surveying. Our postgraduate food security training program is the only one in the world, and we have the widest range of agricultural disciplines at any single South African or African campus. The choices of degree and the options on offer may seem confusing at first. Engineering degrees are more specific, and the options are usually career-focused. Emphasis is placed on training in the specific principles which engineers deal with in their professions. Engineering offers 7 different professional specializations. There are also degrees in land surveying or geomatics and property development. In science, there are structured or career-focused degrees. These include fields as varied as chemical technology, marine biology, industrial and applied biotechnology, and computer science and information technology, to mention a few. There are also more general degrees with a lot of flexibility. In this case, students have the choice of studying a BSc M stream in maths, physics, computer science and statistics. All of these have a strong mathematical component. The other option is a BSc LES stream with a focus on the life and environmental sciences with subjects such as geography, genetics, botany, hydrology, zoology, ecology, and microbiology. In agriculture, there are various different options that involve practical and scientific applications. Students can combine science subjects with specific agricultural orientated. Agriculture and agricultural management are 3-year degrees with a higher practical component. Dietetics is a professional qualification that deals with the role nutrition plays in the promotion of health and the treatment of disease. Most degree programs across the college have field trips and projects that contribute significantly to the curriculum. This ensures that students learn in real-life situations under full field conditions. Many of our lecturers have received Excellence in Teaching awards, are recognized internationally, and are rich in practical experience. Students have access to excellent facilities that rank amongst the best in the world. 300 million rands has recently been invested in new teaching and research facilities, state-of-the-art laboratories, and improved equipment. The 30 million rands new School of Engineering building is the envy of engineering campuses across the country. Agricultural students have access to a 356-hectare research and training farm situated near the Pietermaritzburg campus. This facility provides students with hands-on training and is used extensively as an outdoor laboratory for teaching and research in areas such as animal and poultry science, horticultural science, and agricultural engineering. Adjoining Ukulinga is the Bisley Valley Nature Reserve, which supports a diversity of plant and bird species, as well as some large mammals. It provides a rich learning and research environment for staff and students. Based on the Westville campus, the college possesses unique facilities for research into higher voltage power transmission. These facilities are available in only 3 other places in the world and form part of the ESCOM-sponsored Science and Technology Innovation Park. The Science and Technology Education Centre showcases the college's scientific achievements and promotes the public understanding of science, engineering and technology within the region. The college houses a wide range of sophisticated equipment in its microscopy and microanalysis units, staffed by highly qualified and skilled technical specialists. The College of Agriculture, Engineering and Science prides itself on its graduates who are highly sought after.
Good afternoon, students, and welcome to our last biology revision session. Thank you for joining us today, and I'm sure everybody is almost done learning. Or you should be finished learning actually for tomorrow's exam. I'm sure you can't wait to get it over with. And today we're going to do the second half of your paper 2, which is, generally students like the genetics component much better. You can get to, sorry, I mistakenly muted. You can get pretty good marks in this genetics section in this paper. So let's get straight into it. Naomika, if you can admit the latecomers, please. There's quite a few popping up. Thank you. Okay, I'm going to share my screen with you in a moment. Okay, can everybody see a PowerPoint slide? You can just give me a thumbs up. Okay, thanks, Husna. All right. Okay, so as I said, we're going to be going through the second part of your paper 2, okay, and that is dealing with genetics and inheritance, which is going to comprise of 47 marks in your paper. So it's almost a third of your paper just like the evolution section. That was just a few more marks than this. And then your DNA of life, DNA code of life section. I just ask you to mute once you've entered, please. Okay, thank you. Okay, so let's begin with I'm going to do some questions as we did yesterday, some past questions from previous years, and we can go over a few concepts as we're going along. Please feel free to stop me and ask me any questions, or if you are unsure about any of these answers, you are more than welcome to stop me at any point and ask. Okay, so let us begin with that. Okay, I'm going to start with the genetics section, just so that we can get as much practice with it as possible in our 2-hour slot here. Okay, so these questions are all taken from past year papers, so you can see the level. This is from 2016 to 2020, so you can see the level of questioning that they expect for this subsection of your paper. Okay, and it's very sort of straightforward. There's nothing too complicated about it. So if you've learned your genetics section and you've done some practice questions, I think you'll be perfectly fine for it. One tip to remember is that in section A, you're going to be getting name the biological term, or multiple choice, or matching. And sometimes this has been popping up, this section pops up there a lot, because there's a lot of terminology that you need to know in this genetics and inheritance section. Okay, so I would just revise all your terminology, your definitions, okay, because that can also pop up in section A, okay, from this topic. Okay, so let's start with this question first. Mendel observed some characteristics of the pea plant, which he suggested were controlled by inherited factors. He conducted a series of experiments in which he crossed pea plants with contrasting phenotypes to obtain the offspring of the F1 generation. At first, his crosses were simple and involved only one pair of characteristics. Mendel counted the number of offspring showing each of the variations. His results are shown in the table. Okay, so he crossed a whole lot of different plants with these characteristics. Okay, and these are his results. Okay, so the first question is, give the term for the inherited factors that Mendel is referring to. So here, when it says he suggested that characteristics are controlled by inherited factors, what are those inherited factors called today?
Genetics.
What are they called?
Genetics.
No, inherited factors. What are the things that control characteristics?
Alleles. Genes.
Yes, very good. Okay, so genes. That is our inherited factors. Okay. Okay. A cross involving only one characteristic is called what type of cross?
Monohybrid.
Monohybrid.
Very good. Okay.
There are people who are not in these prisons, but spiritually they are in different prisons.
Okay, can I just ask you to mute, please?
There are people like that I'm talking.
Thank you. Okay, name the female structure of the flower where meiosis occurs.
Ovary.
That's correct. Some people, anybody got any different answers for that? You can also say gynecium or pistule, or some, if you want to put in the specific part of the plant as well. That's also acceptable according to the memo, but ovaries is acceptable as well. Okay. And use the information in the table to give the number of each of the following. Characteristics of pods.
Two.
How many characteristics do we have?
Two.
We have texture and color.
Color.
Okay. And how many alleles for seed characteristics?
4.
Very good. Okay. Because we've got 2 characteristics, so that will give us 4 different alleles. Okay. Give the characteristic that is dominant for flower color. Okay, so we're looking here at flower color. When we crossed a violet and a white, we got all violet. So which color is dominant?
Violet.
Very good. Okay, then recessive for stem height.
Short.
The characteristic that is recessive for stem height.
Short.
Correct. Because tall is dominant, because we see when we cross a tall and a short, we get all tall. So that means it must be dominant over short. Okay, so the recessive is short. Okay, if the individuals of the F1 generation are crossed, how many phenotypes for seed color would be expected in the F2 generation? Okay, so if we're crossing seed color, okay, if the parents taken from the F1 generation were crossed, so they are all heterozygous yellow. If they are crossed, how many phenotypes for seed color would we expect? So if you cross 2 yellow heterozygous, what will you get? You'll get 1 of each of the alleles, right? So that is, so you'll still get yellow and green. Okay. Okay, so that was fairly simple, this question, and that it was quite nice because each one of these things is for 1 mark. So you're likely to get probably 10 out of 10 for this. Okay, so hopefully you get a nice easy one like this, but from 47 marks, you're probably going to get some easy and then maybe 2 or 3 really hard ones that are more in depth, but that was a nice easy one to start off with. Okay. Okay. Let me just come back to this question. Sorry, that's in the next section. Okay. Color blindness, daltonism, is a sex-linked disorder caused by a recessive allele, and it's denoted as X to the power D. Okay. The diagram below shows the inheritance of this disorder in a particular family. Okay, so give the phenotype of senzo. Here's senzo here, and you can see this key. Sorry, it got a bit distorted. You can see the key to help you tell whether it's an affected male or female. Okay, so senzo is a male and he is colorblind, so he is an affected male. Okay, so what is the phenotype of senzo?
X2, XD, X.
No, phenotype.
Colorblind. I mean.
Color blindness.
Very good. So he is a colorblind male. Okay. Then genotype of Thule.
X
There's
big D.
Thule here. So what's a phenotype? Tell me first, what's a phenotype? She is a normal
can see.
female, right? She's a normal female, but these are her children. What can you tell about Linda?
Colorblind.
Right. So for Linda to be colorblind, she's a female. And remember, this is a sex-linked disorder. So for Linda to be colorblind, she needs a colorblind allele from the mother and a colorblind allele from the father in order for her to manifest or to show the colorblindness trait. Okay, so she has to have gotten a colorblind allele from her mother and from her father. You with me?
Yes.
So Tuli is therefore a carrier of colorblindness. She's not homozygous normal. She's heterozygous normal. You with me? Because she carries that colorblindness allele, but it's not showing because it is recessive, and you need 2 copies of it for it to show in females. That is why it's called a sex-linked disorder. Is everybody following? Okay. So the genotype of Tuli is going to be
X.
X big D,
Small one.
X small t.
small d. All right. Describe how Linda inherited colorblindness. Now that's just what we've described here. She inherited an allele from her mom and one from her dad. Okay, or you can say mother or father. Okay, the father is definitely colorblind, so he gave X small d to Tuli, sorry, to Linda. Okay. One colorblind allele. So we should be specific when we're answering these questions. Okay, so explain why there are generally more males than females with colorblindness in a population. Okay, so what do we know? Because this trait is sex-linked, excuse me, and it's recessive, it's on the X chromosome. So males have what 2 chromosomes? What 2 sex chromosomes?
X and Y.
X and Y.
X and Y.
Right. So they only need 1 copy of XD. Whereas females have 2 X chromosomes. So in order for this recessive allele to show, it must be in the homozygous state. So it must be XD, XD, X small d, X small d. Okay. So for males, it's much easier or much more common for the color blindness trait to show because they only need 1 copy. They only need 1 X small d. And then they'll be colorblind. But females, they may have that X small d, but they won't show color blindness because they may not be, they may be heterozygous. They may be X big D, X small d. So they like Tuli, they're carrying the X small d, but they are not showing it, showing any color blindness. So that's why we have a lot more males that are colorblind in a population because they don't need 2 copies of something for it to show. Okay, so if you want to write a nice, neat answer for that, males only have 1 X chromosome. So if this chromosome carries that recessive allele, the male will be colorblind. Whereas females have 2 X chromosomes, so they need 2 recessive alleles to be affected. The dominant allele on the other X chromosome will mask the effect of the recessive if they are carrying it. Okay, so if like Tuli, the female's genotype is X big D, X small d, X big D is dominant over X small d. That's what it's saying. Okay, because obviously X small d is a recessive allele. So it's not going to show in the heterozygous state in females. Is everybody with me on that?
Yes.
Great. Okay, so then Molly married a normal male. Where's Molly? Here you go. She's a normal female, and she married a normal male. Okay. Use a genetic cross to show the possible, excuse me, genotypes and phenotypes of their children. Okay, so I've put that on another slide for you to see. Okay, so this is very important, and I think you must all make sure you take a screenshot or take a picture or anything. Just if you've done this question already, mark yourself. Make sure that this is the format you are following, with it set out in this manner so that you can get all the marks. Okay, if you leave one thing out, you're going to miss a mark, and these things are like 6, 7 marks. Okay, so let's not waste any marks anywhere. Firstly, you need to say what is the phenotype of the parents. Normal female Molly married normal male. Okay, that's your phenotype, and there you're going to get a mark for that. Then what is the genotype of the parents? Okay, and this is called P1, the first parental cross. Okay, so the genotype of these parents are X big D, X small d, because Molly is a carrier. And how do we know that? Because here she is here. So she definitely got X small d from her father, but she got X big D from her mother, because she is presenting as a normal female. You with me?
Yes, miss.
Right. So Molly is X big D, X small d, and we crossing we crossing her with a normal male. So that is X big D Y. Okay, it's a normal male, so he does not have the recessive colorblind allele, so it is big D. Okay, so now we've got the genotypes of the parents. Now we need to cross by meiosis, and we need to write down the gametes of these parents. So what are the gametes? Those are the individual alleles or that are going to be split into your gametes where after you have meiosis taking place. Okay, so X big D is going to go into one gamete cell. X small d is going to go into another one. So those are all your gametes, all your possible cells that could arise from this genotype of parent. Okay. Okay, so your gametes are X big D, X small d, and X big D and Y. Okay, it's very important that you write down your gametes. Once you write down your gametes, then you'll be able to put it into this Punnett square. Okay, this Punnett square now is used to do your actual cross. Okay, to cross those gametes and see what offspring those parents can get. Okay, so this is how you're going to set it up. Okay, so X big D and X big D here will give us X big D, X big D. Then Y and X big D will give us X big D, Y. And the same process for the next one and the next one. Okay, what can you notice here? There's going to be one male that is colorblind because he got the X small d allele from Molly and the male Y allele from the dad. Okay, so once you've crossed your offspring, crossed your gametes, these are your offspring. This is your potential genotypes of your offspring. Okay, in this square, yeah. Okay, so you're going to get 1 mark for correct gametes, as I said, and then 1 mark for correct genotypes. So once you've done this actual cross here, okay, in your Punnett square. Now you have to write that out neatly, and you're going to say F1, which is the first cross, okay? And your phenotype of your offspring are one normal female. Actually, normal, you have two normal females. One is just a carrier. You have one normal male and one colorblind male. Okay, so remember, phenotype is how something looks. So that is the physical appearance of that organism. Okay, so remember, phenotype, physical appearance. Okay, so that is how something looks. So write down your phenotype for that cross, and then you're going to write down your genotype. Now, this particular question is not allocated or hasn't written it down here. They've just given you a mark for it in the actual Punnett square. But you can, in the same way, list these genotypes. So you can just write underneath their genotype, and you can say X big D, colon X big D, Y, colon X big D, small d, and so on. Okay, so you can write out, you can list these genotypes that you worked out in your Punnett square. Okay, so you will get a mark for saying, you will definitely get a mark for your genotypes and your phenotypes. And then here's other possible marks that can come from this question. Okay, so that is for 6 marks. So make sure that this format is what you are going to follow. Okay, every time you're going to do a cross, this is the format you must follow, such that you get your full marks for this question. Okay, does everybody understand that?
Yes.
Okay.
Yes.
Move on.
Miss,
Yes.
can I please ask,
Sure.
maybe if we are crossing a second generation of the female, should we write
Yeah.
P2? Should we write P2 or?
Yes, absolutely. So
If
if
it's
you are taking 2 individuals from this F1 generation, let's say you are crossing this colorblind boy here, and you're crossing this, well, shame, you wouldn't cross a brother and a sister, but you know, this if you're taking two from the F1 generation and you're crossing it, you'll cross this and this female and this male. So your next thing will be P2, and you'll write their genotypes and their phenotypes, and then you'll write their gametes. And then when you're doing the Punnett square, you're going to say F2.
Yes, Miss. Thanks.
You understand that?
Yes, Miss.
Perfect. Okay. Right. Can we move on? Okay. Okay. Then another topic in genetics and inheritance is cloning. Yes, I found this question on it from a past paper. The first cloned animal in Africa, a calf named Futi, was born in Northwest in South Africa on 19th April 2003. No fertilization was involved in the production of Futi. She was produced from a single cell taken from the ear of a donor cow named that. The donor cow had a high average milk yield of 78 liters a day. Cloning allows for the production of animals with desired characteristics. That's the definition. Okay, so some people argue that cloning reduces genetic variation in the offspring with no further genetic improvement. Cloning is an expensive procedure and may not be economical for commercial agriculture. Okay, so according to the extract, state 1 advantage of cloning.
Cloning allows for the production of organisms with desired characteristics.
Very good. Okay. if they asked you for a characteristic, an advantage of cloning that is not in the extract. So give me another advantage of cloning. What's another advantage of cloning? Anybody? No answers?
Better yield. Better yield.
Yes, if you think about it, you don't always have to think about it in terms of mammals, but if you think about it in terms of plants, you can, if you clone a plant, or even in this case, the cow was cloned because it had a very high average milk yield of 78 liters a day. Similarly, if you had to clone a plant that particular plant had very sweet fruit, okay? You're cloning something that you know the characteristics of, okay? You're not going to cross something that you may have variation with, okay? So you will always get that same result, okay? Because you're making a direct copy of it, okay? Okay, and now a disadvantage of cloning from the extract.
Some people argue
It's very
that cloning
expensive.
It's quite expensive, and it
Reduces-
actually
reduces genetic
reduces genetic variation of the offspring. Okay. State why the donor cell was taken from the donor cell that was taken from that and not from any other cow. So why was the donor cell taken from Daisy the cow, and not from any other cow? Because she had a very high average milk yield. Okay, so she had a characteristic that was worth making a direct copy of. Okay. Are you with me?
Yes.
Okay. State why an ear cell was used and not an ovum. Why do you think an ear cell was used? Because we were trying to actually clone this organism, okay? If you had to use an ovum, you were not sure of that, you will not be sure of what is in that nucleus of the ovum. The genetic makeup of that particular ovum. Okay, you're not sure of the alleles there. So what they do is they take a cell on any autosome, any normal cell that's not a sex cell, any autosome from this cow that they know the characteristics of. So they know that this cow yields a average milk yield of 78 liters per day. They take the nucleus from that autosome and put it into an ovum. So they remove the ovum from the nucleus from an ovum, any random one, and they take the nucleus from this ear cell and put it into that empty ovum. Okay, and then they implant, they make that ovum undergo mitosis, and then they can implant that into the uterus of an adult female. Okay, sorry, I just overlapped with the following question. But why an ear cell? Because they know the genetic information that's contained there in terms of milk yield, because they can see it from the cow. Okay, but an ovum has an unknown genetic makeup because it's still just an ovum. So they don't want the nucleus from an ovum.
Yeah.
If they are trying to make a clone or something. Okay. Okay, so briefly describe the process of cloning. Okay, this was not in your study guide. I don't know if it's in maybe a textbook or of some sort. Nevertheless, you might get a question where you do have to think out of the box a little, or it's something that you haven't probably learned or seen in your notes before. But if you haven't learned something like this, then just try and answer it to the best of your knowledge in terms of a logical explanation. Okay, always make sure that it's a logical explanation. Okay, so the nucleus of an ovum is removed, for example, from the ear. The nucleus of the ovum is removed. So in this case, a normal ovum from a cow, and it was replaced with the nucleus of a somatic cell. So from the diploid ear cell. Okay, we need it to be diploid because we're not gonna mate this cow with anything. So the cell must have the full set of chromosomes. Okay, so that is another reason as to why we use the ear cell. Okay, because we need the cell to be diploid. Okay, so the nucleus of a somatic cell or diploid donor cell, that nucleus was then implanted into the ovum. That is empty. Okay, then that zygote is stimulated to undergo mitosis, and that will give us now our morula, then our blastocyst, and then that embryo is then placed into the uterus of an adult female for it to grow further. Okay. If you think about plant cloning, it's a little bit easier. Okay. Plants may be cloned by vegetative reproduction or asexual reproduction or tissue culture and grafting. Okay, where you can just, I'm sure your grannies do this or your parents do this, they just take a snip of a rose plant and then just go and plant it, and then that rose will grow. It's a clone of the parent plant. Okay, so that's much more simple with plants. Okay, so a plant with the desired characteristics is selected, and the vegetative part of the parent plant is removed. Okay, for example, a stem, and it's placed in a growth medium or in fertilized soil, or sometimes they place it in water first and then into the soil so it can actually grow a bit of roots and stuff, and then they plant it into the soil. Okay, so it's much easier with plants. You're just removing a portion of it and replanting it. Okay, and that's going to give you a clone of the parent plant. Okay, so that's pretty straightforward. Is everybody okay with that?
Yes.
Yes, miss.
Let me just check the chat quickly. Okay. Okay, so next question. A flower color, purple or white, in a particular plant species is controlled by two alleles, big D, small d. 4 crosses were carried out to determine which allele is dominant. Okay, so we don't know which allele is dominant, but there is one dominant, one recessive. Okay, so 40 offspring were produced in each cross. The phenotypes of the parents and offspring in each cross were recorded. What can you tell immediately from the first cross? So look at number 1. But the
White.
cross number 1, we crossed the purple
Purple.
parent with the white flowering parent, and we got all purple. So what does that tell us immediately?
Purple is dominant.
Very good. Okay. So the first question is state the dominant color, flower color, and it is purple.
Purple.
Okay. Use cross 1 to explain your answer to 2.4.1. Okay, we just did that. When purple flowering plants and white flowering plants are crossed, all the offspring have purple flowers, which means that the purple allele, which happens to be big D, is dominant over the white allele, which is small d. Okay. Okay, so state Mendel's law of segregation. Just some theory. Okay. Mendel's law of segregation tells us what?
For a characteristic, for each characteristic, a plant possesses 2 alleles which separate or segregate.
Oh, sorry, wait, we can't hear you.
For each characteristic, a plant possesses 2 alleles which separate or segregate so that each gamete contains only one of these factors.
Okay, very good. So each characteristic is controlled by 2 genes, which is situated on homologous chromosomes. So when gametes form during meiosis, the 2 genes are actually separated or segregated. That's his law of segregation now. So they are segregated, enabling a gamete to obtain one of the 2 alleles for each parent, from each parent. Okay. So you can write that. This is something you must be able to learn off because it'll likely come up at some point, and it's just learning of that theory. Okay. The 2 alleles for a characteristic separate during meiosis so that each gamete contains only one allele for that characteristic. Okay, that's another way of putting it. Okay, so use a genetic cross to show how the crossing of 2 purple flowering plants can produce white offspring, as in cross 2. Okay, so when you cross 2 purple plants, how come you get some white? It means that one of these purple plants has to be heterozygous in order for, actually both of these plants have to be heterozygous in order for you to get some white flowers. Okay, so purple, the same layout as you can see, also for 6 marks, same exact layout. The 2 purple parents, that's their phenotypes. Then their genotype will be heterozygous purple, heterozygous purple. Because if you're getting white offspring, it means that one recessive allele had to come from the one parent and the other recessive allele had to come from the other parent. So they both have to carry that recessive allele. So both parents are heterozygous. Okay, so now we're going to write down our gametes. Okay, you just look here, big D, small d, big D, small d, and put it in your Punnett square. Okay, and then you're going to cross these 2, and number 1 and 2, and 2 and 2. Okay, so it'll be big D, big D, big D, small d, big D, small d, and small d, small d. Okay, so you're going to get a mark for your gametes, and you're going to get a mark for these genotypes here in your Punnett square. Okay, so what are the phenotypes that we can see? We can see 1, 2, 3. 3 purple is to 1 white. So we have a 3:1 ratio of purple to white. Let's go back and check if that is what they got in this cross here. Okay. In cross number 2, it was 31 purple to 9 white. So roughly a 3:1 ratio. 9 times 3 is 27. Okay, so roughly a 3:1 ratio between purple and white. When you cross 2 heterozygous parents. Okay. So we had purple is to white for phenotypes. Okay, everybody understand that? Are you able, do you think you can do this? You must be able to do this because they will definitely ask you to do a cross of some sort. And this is the easiest, simplest layout. Okay. Is everybody okay with that?
Yes, ma'am.
Is there anybody who doesn't understand any point of any part of the working out? Okay, let's move on then.
One response was easy but confusing.
Sorry?
One response was the cross is easy but also confusing.
Yes, when you have big D, small d, and also another tip is make sure that you write your letters differently. For example, if the alphabet they've given you is C, make sure you put a little like mark on the C so that you can easily tell which is a big C, which is a small c, because the letters look the same. You with me? So either try and use letters that are different in capital letters and lowercase, or put a little tail on the small c or something like that. If you have letters that are the same.
Or if you have two different color pens, like one black pen and one blue pen, you can also do that as well. It's easier to spot.
Yes, that's also an easier way.
Ma'am.
Okay, so if you write your gametes correctly, then you will get your Punnett square correct, but make sure that you can clearly see the difference between a capital letter and a small letter. Okay.
Okay, there's a hand raised.
Yes. Yes, Keo.
Ma'am, I wanted to ask, in my prelim in Gauteng, we had a genetic cross with blood types. How would you actually write that, or like write it out if like correct?
Okay, so blood types are multiple alleles, okay? And for blood types, I have an example here. I don't know if you have
I'm saying we had a genetic cross. We had a genetic cross, like we were given alleles and a phenotype for those alleles, and also a blood type in it. So we had to do a genetic cross with the blood type, but I didn't get those marks because I didn't understand how I was able to write it.
Okay, so you had two characteristics. You had like a normal height or whatever.
A normal genetic cross, and then with the
blood type as the second characteristic.
Yes.
Oh, okay. No, you can do it. I've never seen that type a question that's got a blood type with a normal characteristic. For example, tall and blood type AB. Is that what you mean?
Yes, that's what I mean. That's what I mean.
Oh, okay. Okay. In that case, I would just, you can do it, but for example, let me do one. Let me show you how it could be written. Okay, let me just do it on a new slide. Okay, if you had to put it, oh gosh, sorry, I don't know where this is gone now. Okay, so let's say you have okay, you can write the height will be big T, big T, big T, and then because it's a dihybrid cross, then so you must put your next characteristic directly next to it. Okay, and what would be your gametes or your genotype for the blood type? Let's say this person is has blood type A. Okay, so that person can be, let's say they are heterozygous for blood type A. So it will be I with a A in superscript.
Mhm.
Okay, and then a small I. Let me put that nicely here. Oh gosh, where's my superscript thing? It's gone. Okay, oh, I can't even see that. Sorry. Let me make that bigger. Okay, so it's going to look something like that. So here's your allele for height for one characteristic, and directly next to it is going to be your allele for
different blood type.
Blood types. Okay. Yes. So, and then when you're splitting it up, you're going to have to go, why is this not fitting?
T and T.
You'll have to go big T, and then you'll have to go big IA,
small I.
Okay.
Yeah.
So it's going to look like that. Okay. So that's one characteristic. That's the other characteristic there. Can you see that?
Okay. Yes, ma'am, I can see it.
Okay. So remember with blood types now, with blood types, they are multiple alleles. Okay.
Yes.
So there's actually more than one allele for a given phenotype. Okay. Okay, and that the nomenclature for that is this. Okay, where you've got an I as the base and then superscript letters. The recessive form is small I. Oh, God. There you go. Okay, so it'll be a capital I with A or B in superscript, and then the next allele will be a recessive form, so that is a small I. So that's how you have to write that.
Okay, ma'am.
Okay.
Yes, ma'am.
But it can be done as this dihybrid cross, but I doubt they'll ask you such a hectic question. But
they did in the prelim.
Oh, is that it's from your trials paper?
Yes, ma'am.
Shame, sorry. But hopefully you got part marks for at least the complete dominance aspect.
Yes, ma'am.
Okay.
Yes, ma'am. Thank you, ma'am.
Okay. Right. So let's move on to the next question. A certain plant species has the following alleles for each characteristic: number of seeds per pod and leaf shape. Okay. So one seed is big P and 3 seeds are small p. And leaf shape, normal is big L and wrinkled is small l. Okay, so the table shows the results of different offspring produced by a genetic cross between two plants of these same species. Okay, so they got 4 different phenotypes from a cross between 2 parents. Okay, so how many genes of the plant are considered here? We've got 2 different characteristics, so that means we are looking at 2 different genes. Okay. Okay. Name the dominant phenotypes of this plant. Dominant phenotypes. So the dominant phenotypes, you can see big P. So we've got 1 seed and a normal shape. It's asking for the phenotypes. So you're not going to say big P and big L. You're going to say what does that look like? So one seed and normal shape. Okay, give the genotype of each parent.
Big P, small p.
Okay. So remember, in order for you to get all these different characteristics, the parents have to be heterozygous for both characteristics, because you've got offspring with recessive and dominant traits for each of those characteristics that we are looking at. Okay, so the parent must be big P, small p, big L, small l. Okay, both of them. Just gonna write it like this. Okay. Give the number of offspring that are homozygous recessive for both characteristics. Okay, so what's homozygous recessive for both characteristics? We're looking for small p, small l, small l. Okay, I just wrote that there so you can tell. So we're looking for wrinkled shape with 3 seeds. How many of that?
32.
32. Okay, that is our wrinkled leaves with 3 seeds. 32 from the table. Okay, so that question was pretty nice and easy as well. Okay. Then the back of a leopard frog can be spotted as shown below, or it can be without spots. Spotted frogs were allowed to interbreed and they produced 150 spotted offspring and 50 offspring without spots. Okay, so let's say a 3:1 ratio of spotted to non-spotted. Okay, just so we can give ourselves some information there. Okay, so which phenotype is dominant just based on those results?
Spotted frogs.
Very good. Okay. Explain why. Because we have a 3:1 ratio of spotted to non-spotted frogs. Okay, there were 3 times more offspring without, the spotted offspring were 3 times more than the offspring without spots. Okay, so the ratio of spotted offspring to offspring without spots was 3:1. Okay, as we said. So then obviously that means that spotted was dominant over non-spotted. Okay, a frog that is heterozygous for a spotted back was crossed with a frog without spots. Okay, so heterozygous, so he was carrying the recessive allele, and a completely recessive partner frog was crossed. Using the letters big D and small d represented a genetic cross to show expected genotypes and phenotypes of the F1 generation. Okay, so your big D is going to represent your spotted, and your small d is going to represent your
non-spotted.
I'm going to name it plain. Okay, just so it's easier to say. I'm getting tongue-tied. Okay, so we know that from the question. Okay, so this is what your Punnett square should look like. Okay, we're going to follow the same layout. Phenotype, you got a spotted heterozygous individual, because they told us that in the question, crossed with a homozygous recessive individual, so no spots. Okay, so that is the phenotype for homozygous recessive individual in this case is no spots. And then their genotype is big D small d crossed with small d small d. Okay, your gametes will be big D, small d, and small d, small d. So you put it in your Punnett square, it's going to look like this. And now you go about multiplying your gametes, so to speak. Okay, so we have big D, small d, and when we join this one and this one, we get big D, small d, and the same with the second two. Okay, so we have an overall phenotype of
50%.
50-50. Okay, so 2 of them were spotted and 2 of them were without spots. Okay, so you can also say a 1:1 ratio, because if you simplify this ratio, it's 1:1, 1 spotted to 1 without spots. Okay, and then the genotype is going to be the same exact thing, but you're going to, instead of saying spotted and without spots, you're going to say big D, small d, and small d, small d. If they specifically ask you to write out that phenotype and that genotype. Okay. Okay, and that again was for 6 marks. So please, as this is the 3rd time now we've done it, so, and there was 3 different papers, so you must know it's definitely something that can come out. Okay, so that's most of my questions for the genetics. So it was pretty manageable until now, I think. Did anybody find that too hectic for them to manage? Because if not, then that means you're likely to get those 47 marks. Just remember, it might also come out in section A, which is your matching and give the biological term. So know your biological terms for this section as well. What is an allele? Give the term for an inherited factor, which is a gene. What is a dihybrid? They may say a cross involving 2 characteristics at a time. Then you're going to write down dihybrid cross. They may say a photograph of an individual's chromosomes taken from a diploid somatic cell in which the autosomes are arranged by size and numbered from largest to smallest in homologous pairs. What is that? A photograph of an individual's chromosomes, and the chromosomes are arranged into the pairs.
Karyotype.
Karyotype. Okay, that's very good. Okay. Let me give you one more.
Jerusha?
Yes.
They just requested that you explain dihybrid cross a bit more. You know, the ratio, the 9:3:3:1. And if you have,
okay.
an example of it.
Oh, I don't have an example on my slide.
Yeah.
But you know, when you do usually get that, when you're crossing something like this, when you're crossing 2 individuals that are 2 parents that are heterozygous for both traits. If I remember correctly, that's when you get your 9:3:3:1. You can always try it.
Okay, I'll just share a screen of an example.
Okay,
so
thank you.
So you can just explain.
I'll stop sharing.
Okay. Okay, just let me know if you can see it. Okay.
It's still loading.
Okay.
Okay, we can see it now.
Okay.
Okay, so yes, as I was saying, if you have parents that are heterozygous for, the one is homozygous for one trait and heterozygous for the other.
Yes. So you have
Yes.
the
So
color
they are,
trait and height trait. Let me just type.
sorry?
I'm just going to type here. It's
Okay, sure.
color and height in this dihybrid cross. That's there. We got red. And the height is tall and short. Okay. So tall is dominant. Short is your recessive, which is designated by a small t. Red is also dominant, indicated by a capital R, while your white is allocated by a lowercase r for recessiveness. Okay, so that's you
Okay.
heterozygous phenotype.
Yeah, thanks, Nimisha. So you can see that both your parents are heterozygous for both those traits. Okay, they have alleles for each of the 4 different types of alleles. So when you cross them, you need to make sure that you not only you must have both traits in that gamete. Okay, so there'll be a big R there'll be an R and a T. Okay, you can't just have the R only. Okay, so you have to have an R and a T for each gamete because we are looking at 2 traits. And then when you break down your each parent into their gametes and you do the cross, you must make sure that you put the same trait together. So the R's and the R's must go together, and the T's and the T's must go together. Okay, so then now you've got a complete offspring with 2 traits. So for example, the first one is, what's the phenotype of the first one?
Red and tall.
Red, tall. Okay, so now once you've completed that Punnett square, now you should go with a highlighter or even whatever color item you have, a red pen or a different color of some sort, right? And then you're going to count now. Okay, you're going to say everything that is red or everything that has a big R and a big T. Okay, then everything that has it's easier actually to do the recessive first. If you highlight all your recessives, you've got small r, small t. Then what has small r's but then is heterozygous for the other trait, which will be small r, big T, small t. Okay, so you can work with your phenotypes when you're trying to do your counting. Okay, for each one, say you can even write out this whole thing and then sort it into a simpler form. Okay, so then that's how you end up with a ratio of a phenotypic ratio of 9:3:3:1.
And the other hint that your cross is right is that diagonally you will find your heterozygous genotype in your Punnett square, right? If you find this heterozygous for both your traits diagonally in the Punnett square, you will know that your cross is going correctly.
Okay, that's a university tip. Okay, so look at your diagonal and you should have heterozygous for both traits along that whole diagonal. And that's when you know you have not made a mistake when you were crossing your gametes.
Okay.
Okay. Is everybody okay with that?
Yes, ma'am.
The person who asked the question, is that okay?
Yes.
Okay. Sorry, let me just start sharing my screen once again. Right. Okay. Okay, so now I'm going to just move on to the DNA code of life. Okay, and I just have a few examples here. Okay, can you see that? Okay, and if you had, then we'll take questions at the end where we can do more examples of anything that you'd like for paper 2. If you need me to also go over meiosis again, for those of you who are not here yesterday, we just did it quickly yesterday, but we have some time to go through that as well. Okay, so let's start with DNA code of life. Also may pop up in your section A, where they're asking you terms or multiple choice or matching. Okay. So the diagram below shows the structure of a chromosome. Identify parts D and E.
Part D is chromatid.
Very good. Part E?
Centromere.
Centromere.
Okay, what's the function of the centromere?
Is to hold chromatid.
It joins your two chromatids, and it is the point at which your spindle fiber attaches. Okay, so how many pairs of chromosomes are found in a normal human somatic cell? This everybody should know.
23.
In a normal human somatic cell. 46. A normal cell. Very good. Okay, so you have normal 46. Oh, sorry, how many pairs? Then yes.
23.
23. Okay, give only the letter of the part that attaches to the spindle fibers during cell division.
E.
Correct. I gave you that answer. Okay, represents a gene. Which letter represents a gene?
C.
Very good. Okay, it's showing that band across the chromatid. Okay, so name 2 organelles in an animal cell where DNA is found.
Mitochondria.
Mitochondria and the nucleus.
And nucleus.
Very good. Okay, very good. Then the natural shape of a DNA molecule?
Double helix.
Good. The process whereby DNA makes an identical copy of itself.
DNA replication.
Very good. Right, so that was very easy. You should definitely get your 10 marks for that. Okay. Then I had another question up here I was going to go back to. This, okay. Yes, we didn't do this question. Okay. Oh, let me do this last. Okay, so identify molecule Y in this diagram here. What is this funny looking thing here?
The tRNA.
Okay, there's your tRNA molecule. Okay, the group of nitrogenous bases Z. What does the tRNA carry?
Anticodons.
Okay. Oh, sorry, it doesn't carry anticodon. it has anticodons. Okay, that are complementary to the mRNA. Okay, if X is the next amino acid required after W, then identify nitrogenous bases 1, 2, and 3. Okay, so here, so here's W's anticodons. Okay, so it ends, it's UUC. So we are going to be looking at for X, it's going to be
UGG.
Yes.
okay. So which part of the mRNA are we looking at? A, C, C. Okay, because that's the next triplet. Okay, so now this is the next triplet on the mRNA, and this X is going to be complementary to that. Okay, so what is complementary to ACC?
UGG.
UGG. Okay, so number 1, 2, and 3 is going to be UGG. Okay, so you must put it in the correct order, otherwise you're not going to get the mark. Okay, because you must read it from left to right, because we can see that the ribosome is moving left to right, because here's that polypeptide coming off on this side. So that means this tRNA is going to be added here, and then the next one is going to be added here. You with me? Okay, then the DNA based triplet that codes for X.
TCC. Yeah. It's gonna be TCC.
Okay, so the DNA base triplet that codes for the same bases. So instead of UCC, we're gonna have
TGG. Yeah, TGG.
Yes. Okay. Okay, remember mRNA has uracil, but DNA has thymine. Very good. Okay. Sorry, this was UGG. Okay, describe the process of transcription. Okay, definitely it's a must-know question. You are definitely going to get asked about this. Okay, so we can just use this point to talk about transcription. Okay, Namisha, do you want to share your screen with the picture now? And I can just go through it from there.
Okay, no problem.
Sure. Okay, so you must know your process of transcription and your process of translation. Those are the key processes in this particular topic. Okay. Okay. We've got a slide.
Everyone can see. Everyone
Yes.
can see. Okay.
Okay. Okay. Okay. So if you look at the first bit on the top of that slide, you can see in blue you've got your DNA. Okay. Then you are making a messenger RNA. Okay, from that DNA template. Okay, so can you see that there's only a small section of DNA that's going to get transcribed into mRNA? Okay, the reason we say it's transcribed is because it's still in that same form of DNA, same language, same base pairs as DNA, except that it instead of thymine, it has uracil. Okay, so it undergoes transcription to produce messenger RNA. What message are we talking about? We're talking about a message for a code for a specific protein. Okay, so transcription occurs when we are changing DNA into mRNA. Okay, so for transcription, thanks, Sims. I'm just gonna refer to my slide once again, and then I'll come back to the slide for translation. Okay, so for transcription, see, they've given you translation in this picture, right? But they've asked for transcription. So just make sure you read your question very carefully. Don't get caught out with that. Okay, so describe the process of transcription. We're talking about making an mRNA molecule from a DNA molecule. Firstly, remember, your DNA is going to be wound up. So we need to unwind our DNA, okay, and actually have a straight template strand of DNA. Okay, so that template strand is now what our mRNA will be made from. So in the nucleoplasm of any cell, there's free RNA nucleotides just floating around, okay? And that is actually going to join together to form an mRNA strand that is complementary to our DNA strand. Okay, so your DNA is a template, which means it acts as a sort of a stencil for your mRNA to be made. Okay, so how does the mRNA know what base pairs to code and whatever? It's got DNA. Okay, so the code is coming from the DNA. Okay, so that those base pairs that's on your DNA, or that sequence of nucleotides is going to be a template for mRNA nucleotides. Okay, so the mRNA now has this coded message for a protein. Okay, so the RNA nucleotides that were floating in the nucleoplasm, they join together based on that DNA template strand. And now we've got a strand of mRNA nucleotides that has a specific pattern which codes for a specific protein that the cell may need. Okay, so once that mRNA is made, it can now exit the nucleus and go into the cytoplasm as a little mRNA molecule. Okay, and when it goes into the cytoplasm, it's going to then attach itself to a ribosome, which is this here, and then it will undergo translation. Okay, so for transcription in this particular question, your 6 marks are coming from what this says. Okay, any 6 of these. Okay, the double helix unwinds, the double helix of DNA unwinds and unzips. So those weak hydrogen bonds that were holding the 2 strands of DNA together are going to break. And this forms 2 separate strands. One of those strands is going to act as a template to form your mRNA. So how do we form the mRNA? There's free RNA nucleotides floating around in the nucleoplasm. So this is within the nucleus. Okay, and the mRNA is going to be created by joining these RNA nucleotides in a sequence that is complementary to the DNA. Okay, then that mRNA is now a coded message for protein synthesis. Okay, is that clear? Does everybody understand that explanation?
Yes, Miss.
Okay, you must be able to give this explanation for 6 marks. Okay. because it comes up all over. Okay. So I'm just trying to change the next slide, right? Okay, so here's another example, slightly easier. Okay, identify, oh, yes, Owami, you raised your hand.
What?
Owami,
Yes. Or
your hand is
Wami,
raised.
please unmute your mic.
Oh, maybe it was a mistake.
Translation.
Okay. Okay, identify the process above. What's going on in this picture?
Translation.
Nation.
Okay. Okay, now identify organelle A. What's organelle A?
ribosome.
Ribosome.
Very good. What's molecule B?
mRNA.
mRNA.
Very good. What's the bond at E?
Peptide bond.
Peptide bond.
Peptide.
Very good. Very good. Okay, just that's another tip. Make sure you know what different types of bondings you have. For example, we just
Hydrogen.
mentioned the weak hydrogen bonds between the two DNA strands. Okay, there's a peptide bond between each amino acid in a polypeptide chain. What other types of bonds do you like?
Hydrogen bond. Hydrogen bonds.
Yes, we said there's weak hydrogen bonds between the two DNA strands. Which other ones?
That's all. Nothing else.
Do you learn about glycosidic bonds?
No.
No.
Oh.
Okay, that's at university level. But we also do this particular topic of transcription and translation. You'll learn about it in similar detail, actually. So don't throw your matric notes away when you come to university next year. Okay, so the bond at E was peptide bond, which is what forms between your amino acids in a polypeptide chain. Okay, so give only the letter of the molecule that carries the amino acid. What carries your amino acids?
D.
C. C.
What is that?
That's the actual amino acid.
TR.
It is tRNA. So your tRNA is going to carry that amino acid. Okay, you can see this amino, here's this would be an amino acid that it's going to carry. Okay. So which letter is a molecule that is copied from DNA?
B.
We just spoke about it.
B.
So what is the molecule that's copied from DNA?
mRNA.
mRNA. Very good. Oops, I'm supposed to give only the letter. So what did we say? That was C.
C.
And this was
B.
B. Okay. So make sure you don't make that mistake as well. Okay. Which letter is a molecule that is the monomer or building block of proteins? So what is the building block of proteins?
T. E.
No.
Where's
Peptide.
T?
E. E.
E is the peptide bond.
D.
Very good. Okay,
Oh.
so what is the monomer of a protein? What is the building block?
Amino acid.
Very
Yeah.
good. So you must know, so it might be easier first to answer the question of what it is and then find the letter associated with it. Okay, so in this case, the monomer or building block of proteins, it's an amino acid. So where's an amino acid? The tRNA is carrying it, so it is D.
D.
Okay. Again, a nice, easy question. I hope you get something like this. Okay. Let's go up to this other question. Okay. According to the table, how many, also let me read this to you first. Okay, the sequence of amino acids in a protein molecule is coded for by DNA and RNA. The table below shows some mRNA codons and the corresponding amino acid. Okay. According to the table, how many codons code for phenylalanine?
2.
1, 2. Okay, that is correct. Okay. Then, what is the anticodon for glutamic acid? If this is the codon, what's the anticodon for glutamic acid?
CUC.
UC.
What?
So
CUC.
lysine.
UC.
It'll be C,
Lysine.
U, C.
E.
Okay. Then, a section of mRNA has the following base sequence and is read from left to right. Okay. Give the DNA base triplet for the last codon on this section of mRNA. Okay, so we're looking at this.
TG.
What is the DNA that's complementary to that?
TGG. TGG.
TGG. Okay, remember it must be complementary to that. Okay, the first amino acid coded for by this section of mRNA. So what's the first?
Aspartate.
What's GAU?
Aspartate.
Aspartate. Okay. Okay, a mutation occurred which resulted in the following base sequence on the mRNA molecule. Okay, so that was the, here was the original, and here's the mutation. Okay, so describe the mutation that occurred. So what kind
AGC.
of mutation? Sorry?
One of the codons changed.
Changed how?
AGC changes to AGU.
Yeah.
Okay. Any other changes? No. Everything else is fine. Okay, so what is that? It says describe that mutation.
One of the nitrogenous bases, cytosine, change to uracil.
Okay, so that is called
Mutation.
yeah. Do you learn the specific types of mutations?
Frame shift mutation.
Yes.
Lethal.
Thank you. That's the type of answer I'm looking for. What other types of mutation do you learn about?
Lethal, harmless.
No, it'll either be lethal and harmless is in genetics.
Oh,
Don't mix that up. Mutations, it can be frame shift, it can be a deletion, it can be an insertion, it can be yeah, that's all I can think of right now. But that is when you're looking at your actual sequence of bases
Mm.
of either mRNA or DNA. Okay, so in this case, we've had a swap, those two, actually we've had a swap where you said, what did you say? C changed
AG.
to
U.
U. Okay, so that is now an actual different
mRNA.
a different triplet that we have there now. Okay, so instead of AGC, we've now got
AGU.
AGU. So it's a different triplet, which is going to code for a different amino acid. You with me?
Yes.
Okay. Okay, so explain the effect that the mutation described in 2.4.1 will have on the resulting protein.
It will code for a different protein. The mRNA will code for a different amino acid.
Okay, but in this case, what do we have? Do we have a different amino acid?
No, it's
In this case, we're lucky. It's
same.
not a different amino acid. It's actually the same. So
Same.
AGC codes for serine in our original sequence. And after that mutation, the new codon also codes for serine. So we are lucky in this case. Okay, there's actually no effect of the mutation because it's coded, that change actually coded for the same amino acid. Okay. Since it is same amino acid. All right. Okay, name and describe the process occurring in the nucleus, which results in the formation, sorry, this went a bit off here, results in the formation of an mRNA molecule. Okay, so that is, so they can give it to you, they can word it like this. Name and describe. So naming, it would be transcription, and describing now. Okay, the process occurring in the nucleus which results in the formation of an mRNA molecule. So if you had to name it, it would be transcription. Okay, and obviously that is a mark that is allocated for the word transcription. Okay, if you just start describing it without naming it, you're definitely going to lose 1 mark. Okay, so transcription is the name of the process. And now if you have to describe it, it's exactly the same as we just did before. But this is from another paper that I've taken, another exam paper. So in their model answer, they expect a slightly different wording, which it's good for you to see because you can obviously describe it in your own words. Okay. So the process is transcription. As we said, the double helix has to unwind to create a template strand. Okay, and to unwind and create that template strand, those weak hydrogen bonds have to break that hold the DNA molecule, the DNA double helix strands together. Okay, then one strand is used as a template to form mRNA. Okay, how does it form mRNA? The free RNA nucleotides in the nucleus will join to form an mRNA strand that is complementary to the DNA. Okay, there's actually in this case also a mark allocated for saying that the process is controlled by enzymes. Okay, so that's how you're going to get your 6 marks. Okay. I'm just checking one thing quickly. Okay, we can come back to it. Can you still see my screen?
Yes.
Okay. Okay. Then we have this example. Detectives were investigating a crime scene and found blood on a broken window. They suspected that the blood was that of a criminal. To identify the criminal, they analyzed a DNA sample from the blood and compared it to that of 4 suspects. So here's your sample and here's your 4 suspects. Okay, name the technique that was used to identify the criminal.
DNA profiling.
Yeah, what is going on here? It is DNA profiling.
DNA profiling.
Correct.
Oh, fingerprinting.
Yes.
Fingerprint.
Okay.
Writing.
Jason, can you stop writing on my screen, please? Thank you. Okay. Who is the possible criminal?
Jenny.
Thanks, Jason, for giving us the answer there. Jenny is the possible criminal, and we'll explain it now. Why is Jenny the possible criminal? Here's our sample, and we are comparing it to each of these suspects. Okay, so DNA profiling is when we take a sample of our DNA and we actually break it down into certain genes or certain base pairs, okay? And we look for the same pattern in other samples, okay? And because every sample or every person is unique in terms of their genetic profile, okay? So whichever matches this profile, that means that this DNA belongs to that person because it is unique to them, their alleles and their genes. Okay, so explain why Jenny is the possible criminal.
Her DNA matches the sample.
Okay, so we can be more specific.
The bars are the same.
The band, yeah, we call them bands.
Bands.
Yes, these are little DNA bands. The bands are the same. Okay, so for every band, it's actually a perfect match to Jenny. Okay, state one other use of the technique of this technique.
To identify biological parents.
To prove
paternity testing.
Okay, so it says one other use. So we can't say criminals because we just did this for
Identify
criminals.
biological parents.
Yes, so identifying parents.
Paternity testing. Paternity testing.
Yeah, that's the same thing.
Paternity testing.
Yeah, that's the same thing.
To identify the dead bodies.
Yes. Determining genetic disorders.
Okay. Okay. So those types of theoretical aspects can easily come out in a 1-mark question like this, or even in where the answer is DNA profiling, it can come out in that biological terms component of your section A. Okay. Okay, and now I just wanted to also, sorry, I should have done this beforehand, before going to this question. I wanted to also go through translation with you. Namisha, I'm ready for the translation part of the slide. Thank you. Okay, so after we've transcribed our mRNA from our DNA molecule, now that mRNA is in the nucleus, sorry, it's in the cytoplasm and has attached itself to a ribosome. Okay, and that mRNA now is going to be translated. Why do we say it's translated? Because the mRNA is in the language of nucleotides, but our proteins are in the language of what?
Amino acids.
Amino acids. So we are going from nucleotides to amino acids. Okay, whereas DNA was also nucleotides. Okay, so when we were transcribing, we were still going from nucleotide to nucleotide. But now we're translating nucleotide of mRNA into amino acid sequence of a protein. Okay, so our mRNA molecule is in our ribosome, and that is now going to act as the template whereby we are going to make a polypeptide chain of amino acids. Okay. Okay, and this also you can easily get asked for about 4 to 6 marks. Okay, so each codon on our mRNA strand, or it's a triplet. Okay, so every 3 bases is going to be a triplet or a codon on our mRNA strand. Okay, and each of those codons codes for a particular amino acid. Okay, for example, AUG codes for methionine, and that happens to be your start codon. Once your mRNA, it's running through your ribosome. Once your ribosome hits that AUG codon, it knows, okay, that is the start of the gene, or that is the start of the protein that, or the polypeptide chain that needs to be made. Okay, so methionine is always your start codon. Okay, then every 3 codons is going to code for a different amino acid. Okay, so that is how your mRNA forms that template from which a polypeptide chain can be synthesized. Okay, so each tRNA now, that is your transfer RNA. Do we need to know the names of the start and end codons? I wouldn't say all, but definitely start is AUG. That's easy for you to remember. But I don't think you need to know the stop codons.
No.
Sorry, I can't really see that. There are only 3. And the important thing to note is that when you have a stop codon, it does not add an actual amino acid. It just adds a water molecule. And that is how it actually stops making a small polypeptide chain. Okay, but I don't think you really need to know the actual codons for stop. But if you can remember AUG, then you must know that that is definitely a start codon. That codes for the amino acid methionine. Okay, so as I was saying, you've got this mRNA molecule now that's in your ribosome. It acts as a template. Template for what? A template for your tRNA to come and transfer amino acids to a growing polypeptide chain. Okay, So your tRNA is your transfer RNA, and that is floating around all around the cytoplasm. Okay, and it will carry a specific amino acid based on the based on its anticodon. Okay, so remember now, your mRNA has a codon, has a sequence of codons. Your tRNA has anticodons. So what does that mean? What's anticodons? It's going to be complementary to those codons on the mRNA.
Oh.
Okay. And since it's RNA, it's still going to have uracil instead of thymine. Okay. So your tRNA has anticodons on it. And those anticodons obviously are specific to a codon. And that codon is specific to an amino acid. So therefore, your tRNA is only going to be able to pick up a certain amino acid because it has a certain anticodon. Okay, so tRNA will pick up the amino acid that corresponds to its anticodon. For example, if your codon is for proline, what is that, CCG, what is going to be your anticodon?
GGC.
GGC. So the tRNA named GGC is only going to be able to pick up proline. So it will go and get your little proline amino acid and come to the mRNA codon, the mRNA strand in your ribosome. And once it gets to the ribosome, then it can now add that amino acid to the growing polypeptide chain. And it does that by actually forming, a polypeptide bond forms between each of those little amino acids that the tRNA brings to the ribosome. Okay, so the tRNA will bring certain amino acids based on this mRNA molecule, based on the codons that's found on the mRNA molecule. Okay, so that is your process of translation. Is everybody okay with that?
Yes.
Share my screen again. Okay, so for these two sections, okay, for your study tips for these two sections, for genetics and code of life, definitely for genetics, you need to know how to do the format of your genetic cross. Okay, you must make sure you've got parent phenotype, parent genotype, gametes, Punnett square, phenotype of the offspring, genotype of the offspring. That is what you must have in your answer in order for you to get all of the marks. Do not lose marks by leaving those things out. Okay, make sure you write P1, F1. If you are crossing two offspring, you need to write P2, F2, and what that generation is. Okay.
All right.
Then you need to know your concepts of inheritance, like co-dominant, completely dominant, or incompletely dominant. Remember, your complete dominance is where you have a dominant and recessive allele, and the dominant is completely, it will completely mask the recessive. Okay. In co-dominant, you have two equally dominant alleles, okay, which means that the offspring will have both those characteristics
Oh.
because they are equally dominant. It's like two male lions. They are equally dominant. Okay. If you have incomplete dominance, okay, incomplete dominance is where the one allele is partly masked by the other allele. Okay, so because there's going to be this partly masking situation, you will get an actual mix. Your phenotype will be a mixture of two traits. Okay, for example, if you have white roses and red roses, and the trait for color is incompletely dominant, those individuals who are heterozygous are going to be pink, which is a mixture of red and white. Okay, because the one allele is not completely masked, it is only partly masked by the other allele. Okay, and your multiple alleles, they are all equally dominant. Okay, but you do have a recessive allele, for example, in your blood types. Okay. And then cloning and genetic engineering, I think you should just know some theory about that. Okay, advantages, disadvantages, how it occurs, why we would do that. Okay, I don't think it'll be out of too many marks, maybe 5 or so marks. Okay, it can also come up in in naming those terms. Okay. And then biological terms, definitely you might get biological terms asked on biological terms for genetics, okay, because you know you've just got so much of vocab there that you need to learn, like heterozygous and homozygous and recessive, and all of those terminologies can pop up in section A, where you asked to write the biological term for a certain definition. Okay, and then for DNA code of life, you should know your process of transcription and translation. Do you do DNA replication at this point?
Yes.
Yes, ma'am.
Okay, so for DNA replication, it's a simple sort of 4-step process, if I can go through that ever so quickly. Where your DNA unwinds, because it's in that double helix conformation. Your DNA is going to unwind, okay? So you've got something that looks like a ladder, okay? And you've got your base pairs that are complementary to each other. So A always pairs with T and C always pairs with G. And you might need to know that between A and T, you've got 2 hydrogen bonds. But between C and G, you've got 3 hydrogen bonds. Okay, just a note to remember. Okay, Letlo, you raised your hand. Do you have a question?
Yes, ma'am. Ma'am, I had a question about in DNA replication. Where do the free floating nucleotides come from?
Oh, it's in the nucleus.
Are they just sitting there, ma'am?
Yes, they're just floating around. They're just floating around in the nucleus.
Oh, okay. Thank you, ma'am.
Just like your mRNA, you know, when you're making mRNA, when you're doing transcription, there's also RNA nucleotides floating around the nucleus.
Oh, okay.
Okay, when you get to university, you'll learn that the free floating DNA nucleotides are called DNTPs. It's a precursor form. Okay, it's not your fully functional nucleotide as yet. Okay, so you'll learn about the details of that, but there's free floating DNA nucleotides in the nucleus and free floating RNA nucleotides in the nucleus to make which are involved in DNA replication and mRNA synthesis or transcription. Okay. Okay,
Yes,
so
thank you,
as we said, now for DNA replication, your DNA double helix unwinds, and those hydrogen bonds now that are between your A and T and G and C, those hydrogen bonds are going to break. So you're going to have 2 separate strands. Okay, so each strand can now potentially act as a template, okay, on which a new DNA strand can be synthesized. A complementary DNA strand for each of those template strands can now be synthesized. So each strand will pick up these free nucleotides from the nucleoplasm, and your A will always pair with T and G will always pair with C. Okay, and so now we end up with 2 new double-stranded DNA molecules. Okay, because each of our original DNA strands were acting as templates. Okay, and then in your last step, your ladder formation of DNA has to now coil up again into its double helix formation, because that is the most stable conformation of DNA in its double helix. Okay, and that is just basically the DNA replication explanation. Okay, but it's important to remember that each of the strands in your original DNA molecule can act as templates for new strands to be made that are complementary to them. Okay, so you end up with 2 new DNA molecules at the end of DNA replication. Okay, are there any other questions that I can quickly address?
Just one on punctual equilibrium. I'll just put a picture up of the 2 types of equilibrium for evolution, and then you can just explain the basic concept of it.
Okay, I think for matrix you don't learn about gradualism, but I know you definitely learn about punctuated equilibrium.
Yeah.
And that I was talking about yesterday, that you need to know the difference between your theories about evolution. Okay, so like for example, you've got Lamarckism, you've got Darwinism, and then you also have punctuated equilibrium. So punctuated equilibrium is more about how fast something evolves. Okay, so it's the speed, it's a hypothesis about the speed at which evolution occurs through natural selection. Okay, so it's sort of an add-on to natural selection. Okay. Nims, can you just zoom the slide a little? I can't really see what's on the bottom there. You can just zoom at the bottom if you want. Okay, so over time, these, okay, in this case, butterflies are going to evolve. Okay, but the process is stating that, or the theory states that in short periods of time, there's a rapid change. Okay, that occurs, that actually causes differences in species that are noticeable. For example, if you look at the first node, you can see a brown butterfly. Okay, and the ancestral butterfly was brown and yellow, right? And then after a few thousand years, it became a sort of reddish yellow butterfly. Okay, and then a few years later from there, it became white and brown, which is now looks completely different. Okay, so it's more of a stepwise change and a rapid change in the appearance of an organism, such that we can now differentiate them as 3 separate species. Okay, so it's a natural selection is still occurring in this time, but it's punctuated equilibrium speaks of the actual speed at which this speciation is going to occur. Okay, so the meaning of punctuated equilibrium means that evolution sort of alternates with long periods of change and short periods of change. Okay, there could be certain environmental factors that, species don't need to change because they are adapting very well in a certain environment. But then an environmental factor will cause something to change or force something to change very rapidly, such that we see evolution happening very quickly, evolution of characteristics happening very quickly. Okay, so that is what it means by punctuated equilibrium. You get bursts of this evolutionary change. Okay, so it's short periods of evolution followed by long periods of evolution. Okay, so something can change its characteristics very quickly in response to the environment, or it may be stagnant. It may stay in that, it may stay looking like that for millions of years because it worked fine. It survived fine in that way. Okay. I don't know if that is a decent explanation for you. I'm not sure who asked that question. But is that a bit more understandable? It's just punctuated equilibrium is just talking about about little bursts of change, evolutionary change that occurs every now and then in time. Okay, so evolution doesn't always have to be a very long, slow process. There can actually be instances where there's evolution occurring very quickly. Okay, and that is the punctuated equilibrium.
Thank you, Miss.
All right. Any other questions? Yes.
Please show pictures of
I'll just
about the foramen magnum, spine, pelvic girdle, prognathous, pallets, and cranial and brow ridges.
So I couldn't get much for matric level. There's a lot of university level evolution of the skulls and stuff. So I managed to get this one where you can actually physically see how the cranium and the brows change over time in primates until Homo sapiens. But as you can see from the distinct size of the actual skull itself, the orbital sockets, they changed over time, which also influenced the brow furrow for the skulls. And as you can see, for some of them, they had a lower upper for the cranium section of the skull, much smaller. It all also depended on the size of the brain, as Mrs. Jerusha said yesterday during our evolution section, right? Depending on the size of the brain also influenced the cranium size. And as you can see, depending on also habitat availability of food sources, your mandibles, your jawline also adjusted, and as well as the dentition adjusted according to the diet of the different organisms. So as you know, for us, our dentition is in an oval shape for jawline, while you have others with more distinct, rather larger canines and in sizes, depending on the actual food source these different species had over the years in the habitats that they lived in. Jerusha, if you want to add anything?
The general trend is that over time, the brow ridge diminished. So you'll see in more ancient species, you've got a very prominent brow ridge and large eye sockets, okay? Because that was obviously adapted. They were adapted to be having to look out for predators. Their eyes were much larger, okay? And having their skull go over their eye sockets like that was more of a protective feature. Okay. And also, I think I've read before that the prominent brow ridge also helped for shading their eyes and protecting their eye sockets, really.
Yes.
And so the trend is a decreasing brow ridge or diminishing brow ridge over millions of years, and an increasing cranial size, which means that we developed a larger brain, which means that we became more cognitively advanced over millions of years. Okay, so the cranium is a depiction of how big the brain is because it fits in that spot. The foramen magnum is where your spinal cord fits into your skull. So that's indicative of whether something is quadrupedal or bipedal. Okay, so if it is directly underneath your skull, like we are bipedal, so we can walk upright. But a dog, for example, has their foramen magnum right on the edge or on the side of the skull. Okay, because its spine is horizontal. Okay, so it is bipedal. It's walking on 4 legs. Okay. A chimpanzee, you can see, is in the middle of a dog and a human, right? Why? Because it can walk short distances as using only 2 legs, and it can also walk on 4 legs, on 4 limbs. Okay, so it has that option. So they have that foramen magnum in between. Okay. Any other explanations anybody needs?
The only other one I could get was the pelvic girdle.
Oh, the pelvic girdle. That is in relation to whether something is bipedal or quadrupedal. If something is quadrupedal, then that means it's going to have a narrower pelvic girdle, because the pelvic girdle doesn't have to hold the weight of the whole upper body. If something's walking on all 4s, the weight is distributed on the front legs and the back legs. You with me? So the weight is not on the pelvis. But if somebody is walking straight upright, all that weight from the upper body is directly on the pelvic girdle. So it needs to have a wider center of gravity so that it can support the whole upper body. Is that understandable?
Yes.
Okay.
All right, so that was what I could get to show.
Oh, it's perfect. Yeah. Okay, any other things, questions anybody else has? Okay, all the best to everybody.
Hello, Miss.
Yes, yes. Do you have a question?
I think it was no, miss.
Oh, oh, okay. Okay, thanks everybody, and I hope you all do very well tomorrow in your
Good
paper.
luck, everyone.
Thank you, Miss.
All the best.
Thanks.
Thank you.
Thanks.
Benji, sleep.
Part of the UKZN experience is the opportunity to be part of a community and to network with not only our world-renowned academics, but with future game changers and leaders. They do say that the extent of one's success can be attributed to those they rub shoulders with. Therefore, you must surround yourself with like-minded people. So, how do you become part of a community that is committed to your self-development? How do you become part of UKZN? First, you have to apply. All applications for undergraduate studies at UKZN are processed through the Central Applications Office, CAO. This is for both your course and student residence application. The application process is as follows. Step 1. Consult the CAO handbook. This handbook provides a list of all qualifications available at UKZN. Step 2. Have a look through the various courses offered and choose a program that interests you and for which you meet the minimum requirements. As one of the top 5 universities in the country, UKZN promotes academic excellence. So meeting the minimum requirements does not guarantee you a place to study. Step 3. Find the program code linked to your chosen qualification. This is the code you need to enter in your application form. Okay, let me show you something. You see, a code has 3 parts. Example, for BCom in accounting, the code is KN-P-BCN or KN-W-BCN. Crazy code, right? Well, basically, the KN stands for UKZN, the middle part stands for the campus of your choice, P for Pietermaritzburg, W for Westville. Then BCN stands for the actual course. Make sure to get these codes right. Step 4. Use this program code when completing your CAO form, which is available from the CAO website. Your application will be sent to UKZN. You can always check the status of your application using your CAO number, ID number, or passport number on this website, www.caocheck.ukzn.ac.za. For more information, visit www.cao.ac.za. It's that easy. Pay close attention to closing dates for qualifications. For example, MBCHB closes on the 30th of June, whilst other undergraduate programs close on the 30th of September. What are you waiting for? Apply now and shape your future today.